Today, in order to give you an idea about how this budding research and development field is developing, I've selected some interesting examples. And what this slide shows you is the world grain production per capita. And what you can see here, dating back to the 1950s, there was a dramatic steep rise in this food production. This is referred to as the Green Revolution, which happened because of breeding new species of crop plants, using of pesticides, herbicides, etc. But as you can see here, in the last 15 or more years, this has leveled off, and also there's some fairly large fluctuations here. This is considered a problem by agronomists for several reasons. One of them is that the world population continues to grow. Another reason is that as developing countries become more developed, the per capita use or consumption of agricultural foods is increasing. That's a well-known fact. And another problem is also these large fluctuations. Often these correlate with major droughts that occur in parts of the world and are accompanied by starvation. And we can look at this situation also from another perspective related to water and water availability here. In 1995, about 500 million people worldwide suffered from scarcity of fresh water availability. Based on work at the World Watch Institute, this number is predicted to increase to about 2.8 billion by 2025 and 4 billion by the year 2050. Why are these numbers increasing so dramatically? Well, in part that is because the world population is growing, but it's also because we have been borrowing on time and using a lot of fresh water, more than perhaps is naturally recycled. And I want to show you a quote here from Sandra Postel, who's Director of Water Policy at the World Watch Institute. During the last 3 decades, the depletion of underground freshwater reserves has spread from isolated pockets of the agricultural landscape to large portions of the world's irrigated land. And sort of jumping down to the bottom here, groundwater overpumping may now be the single biggest threat to food production. What is the solution to this problem? And as this slide illustrates, the solution is as clear as June gloom, or I would also like to see this slide as showing that the solution to these problems obviously aren't just coming from one source. There are many things that need to be done, and biotechnology is one of these roads that can contribute. And because of the severity of these problems, none of these stones obviously should be left unturned. Now, as I mentioned earlier, water use has increased dramatically, and this shows you over the past century how water use worldwide has increased, with a continuing steep increase into this century. And agriculture makes up about 2/3 of that water use worldwide. So why is it that plants and agriculture need water? Well, all forms of life need water, and plants need particularly a lot of water for a simple reason, and that is that plants have these microscopic holes on the surface of leaves called stomata, or stomatal pores. And plants lose about 95% of their water by evaporation through these pores. So you might ask, well, why are those pores there? Well, the pores are there because they're necessary, because plants breathe in CO2 from the environment, from the atmosphere, through these pores, and then via sunlight and photosynthesis, carbon is fixed and plants produce and release oxygen. So the CO2 needs to enter the leaves for photosynthesis, and that's what the pores are for. Now, because plants lose water through these pores, that's of course a problem. But as you know, not all plants are equal. Some plants, if you forget to water them, or if you don't water them, or if they don't have water for a while, they wilt very rapidly, while other plants do fairly well. So in other words, these pores in the leaves are regulated. They can adapt. And in a plant that's more drought tolerant, as you can see here, what might happen, we are now looking through a microscope at these stomatal pores in the leaves. Each pore is surrounded by 2 specialized cells called guard cells. And if a plant is more drought tolerant, in response to drought stress, it will close its pores, as you can see here. However, some plants are not adapted to dry regions, and particularly agricultural plants, which were derived from certain areas of the world, might not at all be adapted to responding to drought. Of course, today around the world, we plant these plants all around the world because we need to produce grain and other foods. Now, how do plants close these stomatal pores during drought? During drought, they produce a hormone called abscisic acid, and abscisic acid then closes these stomatal pores. So one of the main thrusts in my laboratory, and also in a handful of other laboratories around the world, is focusing on asking, how do these guard cells perceive the hormone signal, abscisic acid, and how do they then close their pores? And the answer, as you might imagine, is fairly complex, and this slide illustrates an example of the type of findings that we are coming up with. This shows you a map of the New York or Manhattan subway network. And similarly, when we look at signal transduction by a hormone, say abscisic acid were a hormone that you apply to the guard cells here, and if this red line running from up here from Columbia University down to Brooklyn were the response pathway, and once the train arrives here, the stomata close, you can see that there's a complex network of events that occurs along the way. So you could think of a drought-tolerant plant as being the express train that makes only few stops and arrives rapidly down here, and the stomata close, while those plants that are more wilty when you stop watering are the local trains that make a lot of stops along the way, and the plant loses its water and wilts, dries out, and dies or is damaged before it makes it to the final destination. So sort of with this analogy, our thought a few years ago was that if we took this local train and if we deleted some of the stops along the way, or in scientific speak, if we deleted some negative regulators in the pathway, we might be able to speed up the process. And we've made some progress with this only in the last year or two, or a little around in that time frame, and I want to show you some examples. This is here a mutant plant in which a single gene is deleted, and these plants have not been watered for the last 12 days when this photograph was taken. You can see here in the mutant plant, these so-called rosette leaves down here remain green, turgid, and full of water. While the wild-type plant, these leaves are hardly visible anymore. They have wilted, turned brown, and largely disappeared. The reason is that the hormone abscisic acid can more rapidly close the stomates in this plant and conserve water over a longer period of time. This plant can survive longer in waiting for the next available rain or watering to occur. Now, this slide shows another example that we're working on in the lab. This is in collaboration with a laboratory in Korea of Hong-Gil Nam and Jun Kwak in my lab. Again, a wild-type plant that has wilted. In the transgenic plant in which the expression of a single gene has been repressed, you can see that this plant is not wilting as much. Recently, we've discovered a third gene, or if you will, a third subway station that we can take out of the pathway, and that's work of Veronique Huguevieux in my lab. And the mutant is shown here, and this is the wild-type plant. This is from a Columbia ecotype. That's why it's labeled COL, or COLO. And these plants have not been watered for a few days. And if you look closely, you'll see how the wild-type plants, these leaves here called choline leaves, or the rosette leaves, are beginning to wilt, and even the flowers are beginning to droop. While in this mutant plant, in which a single gene has been deleted or knocked out, the plants retain water because they close their stomates more effectively and more rapidly in response to ABA. Now, what's interesting in all three of these examples, we have actually not added a foreign gene to the plant, but we have, as I mentioned earlier, turned off individual genes that can be thought of as negative regulators in the signaling pathway. Now, obviously, my laboratory is interested in why do these genes and mutations do what they're doing, and that's where we have developed an expertise to characterize exactly where, why, and how within this subway map are each of these genes acting. And they're not all the same. They're different, and we find very interesting results in this regard. But it is exciting also for me because when we started working on this more than 10 years ago, this was one of our goals, and now we can start seeing the light that it is actually possible to modify plants' response to drought. I'd like to talk about another issue related to water and plants, and that is in order to produce enough plants for food around the world for thousands of years, irrigation has been used. And irrigation is very important in California also to produce the agricultural produce in this state. Obviously, we use Colorado River water and irrigate quite a bit. And agriculture is about the second largest industry in the state with an output of over $25 billion a year. Now, that's all very good, but at the same time, as you might know, the Colorado River water is fairly high in its salt content. So over successive generations of irrigation, the water is used by the plants, the water evaporates, and what remains is salt on the ground. And this is a big problem not only in California, but in many parts of the world. In fact, in about 1992, it was estimated that about 42% of the irrigated land worldwide was affected by salt stress, the remaining salt that remains in the field after, say, several generations of watering with this water. This slide here shows you a close-up view of such a salty crop field, and as you can see here, how the salt residue builds this white crust on the soil. Plants do not like sodium chloride. When they take up sodium chloride, it's toxic, and that reduces crop yields. But also this type of salty fields, these type of salty fields that are occurring is also leading to other effects. For example, in some parts of the world, such as Indonesia and other parts, when a farmer has such a field, he needs to create new land, and often the way that's created is through slashing and burning of rainforests or other natural habitats that then need to give way for new agricultural fields. So it is a big problem, and it's a problem that is increasing. Today, about 10 years later, after that slide I showed you, it's estimated that 50% of the world's irrigated land is affected by salinity stress. This slide here shows you a rice field growing in an area with fairly fresh water that is not under salt stress, and this shows you a rice field that's exposed to increased salinity, increased salt, and you can see the difference in the yield that is occurring here. So what is the solution to this problem? This, again, there are many solutions and many things that are being done. One of the possible solutions that is being explored and where there's a lot of hope, particularly among farmers who face these problems, is also biotechnology. There are not yet applications in the field, but this slide here shows you work of a colleague at the University of Toronto in Canada, Eduardo Bloemwald. What Bloemwald and colleagues have done, as published about 2 years ago, is they overexpressed a membrane transport protein in the right plant here. And this transport protein removes sodium from the cytoplasm of the cells as it enters the cell. Now, both the wild-type plant and the mutant here were watered with a high salt content solution, 200 millimolar sodium, and you can see how the wild-type plant is not doing very well, while this plant is doing a lot better. Now, this is a gene that has been discovered that's important for dealing with salinity. There are a number of other genes that might enhance the ability of plants to survive under salt stress, and there are a number of laboratories working on this. And this is at a stage of gene and mechanism discovery, as you can see here. So the examples and the topics that I've talked to you about today are examples where research and laboratories like my own and many other laboratories are working on these problems and discovering the pathways and the mechanisms that are involved in how plants can deal with stresses such as drought and salinity. But I also want to mention a few examples, that already today can be found in the marketplace and where applications have already been developed. One of these that we read about is plants that have been engineered to be more resistant to insects. As you know, insects cause a lot of detrimental effects in plants, and in order to deal with this during the Green Revolution and today, we spray a lot of pesticides on these plants that kill off the insects. Usually these pesticides are fairly broad and they kill off pretty much every insect that you see if you go out there in a field. I was talking to Matt Whitman, who together with his brother Bill runs a farm up in North County, San Diego, Escondido, and he says they have to spray about twice a week and every darn insect that's out there pretty much dies, not just the insects that attack their crop. So Matt was excited about some of the new technologies that are being developed, and one of these is that corn or cotton or soybean, for example, expressing a single protein called Bt protein, which stands for Bacillus thuringiensis protein, allows that plant to be more resistant to the insect that starts eating, say, the corn cob. As it eats this, that protein that is expressed is toxic to that insect, and then the insect dies, while the need to spray all these pesticides in the environment is reduced. Now, there are also other examples that have been developed, and another important problem in agriculture is weed growth. There are weeds that grow, and the way we've dealt with this, starting with the Green Revolution, is to spray herbicides. Some of them are toxic and end up in our groundwaters, and a herbicide was developed by companies. The herbicide, or the market name for this, is Roundup or Roundup Ready. So many of you have heard about this. You might be using it in your yards and gardens. And the special thing about Roundup is that it's a substance that degrades very rapidly to inert or inactive non-toxic elements after you've sprayed it within a short time. That's the important property of Roundup. So companies have developed agricultural plants to be resistant to this Roundup, so this can now be sprayed. And this also obviously has beneficial results for the farmers that need to buy less such herbicides and spray less herbicides, and it has clear beneficial properties to the companies that develop these seeds and sell them to the farmers. But it also has beneficial effects for the environment. As we can see in this slide here, these type of soybeans that are resistant to Roundup, to the herbicide Roundup, have been in the marketplace for the last 5 or 6 years or so. And what you can see here are herbicides that are under groundwater advisory labeling, that are dangerous to our groundwater, and how dramatically already in the last few years, 96, 97, 98, 99, the use of these herbicides has declined dramatically because of use of such soybean plants. Now, at the same time, obviously, this curve here has not yet maxed out because only a certain percentage of croplands are planted with these plants. Down here, I don't know if you can read it, but these are some of the herbicides with groundwater advisory, and they have quite interesting chemical names like bentazone, alachlor, dimethanamide, etc. So we can see that there's a reduced need for these herbicides, and that certainly would appear to have some beneficial effects to our environment over the long term. I want to mention another discovery that was made by Roger Beachy and colleagues, and this is that viruses are a dramatic problem, particularly in tropical regions of the world. So plant viruses attack plants, and a problem with viruses is there's no real chemical way to kill these off. But what Roger Beachy and colleagues discovered in the mid-1980s in a laboratory was that by expressing a single viral protein in the plant, you can make the plant more resistant to a virus. And this is an example here of an experimental field in Hawaii in which papaya plants expressing this single viral protein, the plants shown in the middle, the greener part of the plot, are expressing this protein, and you can see how they're green. Now the lighter green plants here are the wild type plants, the non-transformed one. And if you look here at a close-up, on the right you can see the leaves of these virus-resistant plants, and on the left you can see that these plants are close to dying, and they have very few leaves, as shown here. Now, this technology does not just work in viruses. It can be applied to many other tropical plants. And viruses are a big problem in the tropics, in Africa, South America, in the Caribbean, in Hawaii. However, there are also some ironic and interesting aspects of this work. In Hawaii, farmers produce papayas because they'd like to export these to the Japanese market, because Japanese people like papaya quite a bit. But in Japan, the acceptance of such a transgenic crop has not yet been reached, and obviously there's a lot of important work that needs to be done in speaking and informing people about what the meaning of these transgenic plants is. But there is a lot of promise here, and particularly, I think, important promise for countries like Africa, where staple crops like cassava that are important for producing enough nutrition for the people working in those countries, that technologies are being developed to make cassava more resistant to viruses, as well as other plants. So this is an exciting advance that's happening at this time. This slide illustrates another advance that has been made here at UC San Diego in the laboratory of Martin Crispiels. And that is that there are not just problems in obtaining enough yield from your crops, but there are also so-called post-harvest losses that play a particularly important role in the developing world. When we harvest seeds and grains, these are peas shown here, we usually store them in a way that we won't have insects and bugs that start eating holes into them, bruchid beetles and other things. However, in the developing world, the storage of seeds is not as technically advanced, and there are substantial losses that occur through bruchid beetles, for example, that start chomping away at these products. So work in Martin Crispiel's lab in the laboratory has shown that by expressing a single gene called an alpha amylase inhibitor in these plants, that the ability of these beetles to munch through these yields is greatly reduced. Now, this is an example, as well as some of the other examples that I told you about, that isn't in the marketplace, but this is happening in laboratories. And obviously, with each of these discoveries, one has to ask, does this have any effect on humans? And various safety and regulatory networks have been developed and are continuing to be developed in order to address possible questions with each of these products. It is interesting today that obviously we and about several 100 million other people around the world have already been consuming corn and soybeans that are resistant to resistant to insects and resistant to herbicides over the last 5 or 6 years. And to date, there isn't a single case that's known that this has had any detrimental effect to humans. So that's a fairly long time without seeing many detrimental effects. But in each case, one needs to ask this question, obviously. So if we take a step back from this work and ask, where are we today and where are we going? For example, where are we in this discovery of addressing food concerns as well as environmental concerns in the new century? I would say if we compare this to the revolution we've seen over the last 40 years or so in microelectronics, that we're probably at the stage of where the first prototypes of transistor radios were being developed in the mid-1960s, 35 years ago. That is, we're seeing some first products that are being developed and some promises from research and laboratories, as I've showed you today. But it's really difficult to predict what all the possible applications will be in the future. And I think if you were sitting here in 1965, it might be hard to predict the effects that the microelectronics revolution has had on us today, whether it's computers, the internet, cell phones, etc. So it's a very exciting time. I wanted to actually address another subject, and that is the following, that not only are plants being considered today for improving agriculture and effects on the environment, but plants are also being considered to be used as green cleanup tools in contaminated sites. And in the US, we have many such contaminated sites. They are referred to by the EPA, Environmental Protection Agency, as Superfund sites. And what this list shows you here are some of the most toxic elements found at these sites. The list is obviously a lot longer, and the estimated cleanup cost for these so-called Superfund sites is on the order of about $750 billion over the next decades or so. Some of the most toxic elements shown here in red are metals or metalloids. Now, what's the problem with metals? You can't degrade a metal in the soil. So you need to remove those metals, and that's part of the reason that this cleanup is so expensive. So what is the technology that's used today to clean up such sites? That's illustrated in this slide. The way this is done is you dig up that soil, you load it on a dump truck, you drive out to the desert or some dump site and dump it again there. So you move it from point X to point Y, and that obviously is very expensive. Now, it's been found in research and also by ecologists that some plants are particularly good at taking up heavy metals out of soils and transporting them into their shoot system. And it's been predicted that this could be really good for removing metals from soils because the root system permeates soils, and it's been shown in some field trials that this actually works, that plants, after several generations of planting, can reduce the levels of toxic metals in soils to below those levels considered toxic by the EPA. Now, in order for this to work, one would want plants that produce a large biomass, that grow rapidly and grow very large. But most of the plants that have been discovered that are so-called metal hyperaccumulators are plants that are fairly small. So one of the goals of research in my laboratory, but also in many other laboratories around the world, is to discover mechanisms by which these hyperaccumulator plants take up heavy metals, move them into shoots, and detoxify them. So what would one do if plants, or what does one do when plants take up such metals and accumulate them in their leaves? One could then harvest the shoot parts of the plants, or concentrate these metals through ashing, and some people have suggested even regaining or reconcentrating these metals for use in industry. But in any case, in order for this to happen, a number of genes need to be discovered that play important roles in this process. In my laboratory, we have cloned and identified some of the first heavy metal transporters through which plants can take up heavy metals. And we've also found a mechanism, an important enzyme that plays an important role in detoxifying heavy metals in the plants in order to help these plants accumulate more metals. The thought among the scientists working in this field is that if we want to make so-called heavy metal bioremediation a reality, one probably has to engineer plants with a number of different genes to maximize the efficiency by which plants can remove such metals. And there are quite dramatic advances being made here in research and also in some biotechnology companies that are starting to stick various genes together towards this goal. I'd like to leave you with a quote from Norman Borlaug, who is the father of the Green Revolution, and now today is looking at the promise and the problems that exist in our future. It took some 10,000 years to expand food production to the current level of about 5 billion tons per year. By 2025, we will have to nearly double current production again. This increase cannot be accomplished unless farmers across the world have access to new biotechnological breakthroughs that can increase the yields, dependability, and nutritional quality of our basic food crops. So with that, I'd like to thank you for your attention and for coming here today. Thank you very much.